Monoclonal antibodies and rabies infection. Title may sound unique, but there's a lot of takeaway in this presentation. It's actually my experience as well. Almost always when we enter a PhD program we get assigned an advisor. That didn't happen to me. It was the first takeaway. This independence and my background was very important to design my study. I should mention it was Professor Talwar's confidence in me, though he was worried I didn't have enough technical support, but his weekly updates on my progress this project was possible. I could have failed, but Prof's confidence in me being a veterinarian, I knew I was assured of a career. Monoclonal antibodies. I was fascinated with this technology from mid-80s, but I never had a chance to think about it, between my job as a veterinarian, managing a poultry farm and private practice. As soon as I finished my BVSC, which is a long 6-year course, interspaced with a year of farm management. Knowing one's background helps in setting the future. We study physiology, microbiology, pharmacology, pathology, gynecology, obstetrics, breeding, genetics, surgery, medicine, so on. This includes clinical rotation starting from 2nd year. I have to describe what we go through in this detail, because most often there's a misunderstanding. There's a thinking all bachelor's or master's degrees are the same. This happens even by notable institutions like National Institute of Immunology. If you look at their ad for PhD program, it becomes evident. Even today I get numerous calls. The hiring managers are focused on a particular technology. On looking their LinkedIn profile, they describe various places and positions they held. Very rarely they talk about their future plans. One hiring manager talked about was ELISA. At Charles River they talked about HPLC, another manager about PCR. What happened at other companies like Pfizer, Bristol Myers Squibb, Amgen, Thermo Fisher, Sanofi, so on was sad. Although I worked at American Red Cross for 20 years handling at least 1,000 units of blood and platelets every day. In one company they were handling platelets. I suggested not to get involved at once. Instead start a small facility as a trial. But my suggestion did not hold ground. It was a new facility. Keeping idle was not my liking. I even suggested 10 different topics for informal discussion. In one place I said these brick and mortar buildings will be a thing of the past in 20 years' time. I did realize most of these managers are basic science graduates. Their background did not help them to expand their knowledge. We have to start thinking of remote analysis and compiling. I strongly believe it's the direction future pharmaceutical industry is heading to. Needless to say, what happened? I was let go. Yes, I had a stroke, and I am still limping and can't use my right hand to even sign. But I was able to drive and used analytical balance and operate HPLC both in the lab and remotely. My disability in lugging my backpack 3 floors to the lab was an eyesore. This I expected. I was let go by all these places. This is a very important takeaway. At no instance I gave room for negative thoughts. If I did, there's no end to it. I still work at Lowe's and another food store as a cashier. I need the money, if possible, to buy used analytical instruments. I am starting an analytical chemistry lab in my basement and built a shed for that purpose. Get all like-minded colleagues to remotely participate and share their expertise. Again, I was firm in my commitment back then to develop a recombinant rabies vaccine. It was possible because of my veterinary education and my experience at IVPM. I knew I didn't have a clear path to vaccine development, but I had the urge to accomplish something significant. True, there were many unanswered questions. Most of us finish BVSC and practically the very next day we are offered a job in a veterinary hospital. Few of us get to serve in a place called Institute of Veterinary Preventive Medicine. I have traveled both in US and in India. I think this is the best place for any veterinarian. There we make all sorts of vaccines for cattle, sheep, goat, pigs, and poultry. On top of it we also make reagents for diagnosis of many diseases. I was placed in foot and mouth disease epidemiology section. Our center produced foot and mouth type C specific anti-serum and complement in guinea pigs. Yes, foot and mouth disease is still endemic in India. It was eradicated in Western countries, sacrificing all cloven-footed animals. At IVPM we get informed when any male calf born in our farm. There's a different procedure to care for the newborn for the first week or 10 days before we sacrifice. On the day of sacrifice both Dr. Ahmed and myself collect blood to prepare newborn calf serum. We also collect calf thyroid and kidney to prepare primary cell monolayers to grow foot and mouth disease virus field isolates. Simplicity was core to the activities of the Institute. We made our own tissue culture media, basically it's phosphate buffered normal saline mixed with lactalbumin hydrolysate, yeast extract with 10% newborn calf serum. This type of background and experience helped me all through my future. We also get to do safety and potency studies of all the vaccines we produce. It was like FDA requirement of SISPQ. After graduation in 2 years at IVPM, I did masters in virology and immunology. Then I traveled 1,500 miles north to join National Institute of Immunology at New Delhi. It was a new institute and buildings were coming up. They were temporarily functioning at All India Institute of Medical Sciences. At the interview, the director asked me what you know about immunology. I said, recognition of self and non-self. He was very pleased with my answer, and he thought it was a short and crisp answer. Since he knew I am a veterinarian, he asked me to come home with him and have a look at his dog. Right then I knew I was selected, and a month later joined NE. My project was to produce human monoclonal antibodies as a treatment for rabies infection. I rejected the idea on that day itself. Anyone who knows about rabies will agree with me. Monoclonal antibody is not a good idea. Maybe for diagnosis, yes, but certainly not for treatment. I will come to it later. But at NE we were having problems even with mouse monoclonals. Though tissue culture was not new to me, hybridoma was. Then I realized others also had the same problem. They have made hybrids with the same myeloma cells before, but not now. They changed everything, media, HAT, FCS, PEG, even water. But I had my suspicion about mouse myeloma cells. About the same time, I got to know two people. One Ms. Danuta Kosbar from Wistar Institute, Philadelphia. She has made human monoclonals from human peripheral blood by EBV transformation. Her advice, human monoclonals as a research project is okay, but for PhD it's risky. She was right. Even today after 30 years we are having difficulties. Another person I got to know was Mr. Dick Skull from Netherlands Cancer Institute. He agreed to send me some new sp2/0 cells, which he did. I also got some cells from Dr. Mohan of AIIMS. Finally we were successful making mouse hybrids with the new cells. Since I am a veterinarian and knew about rabies and have observed both animals and human patients, certainly monoclonal antibody therapy may not be helpful for a sure fatal disease. To my knowledge, there's no treatment for the infection. I was dreaming of a recombinant rabies vaccine. It was all in my mind, but I didn't know when and how to start. Meanwhile, I collected few mice brain samples infected intracerebrally with low egg passage, rabies virus brought from IVPM, Ronapet. Spleen cells harvested from these mice were used in making hybridoma. No firm protocol of making a vaccine was set. There was a paper published in 1984 in Science of expressing rabies virus glycoprotein analogs in E. coli, but it did not protect immunized mice. I guessed lack of glycosylation in E. coli was the reason. Around the same time there was another paper in Nature from B. Moss's lab expressing HTLV3 glycoprotein gene using Vaccinia virus as vector. I was hoping if only I had access to this system, it would solve all my problems. Expressing is one complexity, administrating the protein as vaccine is another challenge. Both will be solved using Vaccinia virus as vector. I already had experience with pigeon and fowl pox at IVPM. My hope was the Vaccinia virus behaves like other pox viruses. By the time there was another paper of oral vaccine using raccoon pox virus. Slowly my confidence increased that I was in the right direction. Now we solved mouse monoclonal issue. Making murine monoclonal had been established. Now hybridoma tested reagents are commercially available as a kit. It's fusion of spleen cells with mouse myeloma cells in the presence of PEG. Screening for positive hybrids is usually by ELISA. There comes a stage when producing hybridoma known as limiting dilution technique. Means selected positive hybrid is subcloned by diluting the clone and plated in a 96-well plate so that each well gets less than one hybrid cell. Meaning every 2 wells should receive 1 hybrid to make sure progeny from a single cell. True meaning of monoclonal. There will be a layer of mouse peritoneal macrophages as feeder cells. To To make large quantities of the antibody we produce ascites in mouse. Pristine primed mice will be injected with monoclonal to produce ascites. Whole procedure may take 4 to 5 months. Hybrid clone gets established and produce ascites. After a certain period, we get to tap peritoneal fluid, probably 4 to 6 drops. Each drop contains large quantities of antibody. Probably 2 or 3 times we can collect by the time mice will be in extreme distress we get to sacrifice. Human monoclonals. This was my primary PhD project. But I gave up the project on the very first day itself. Rabies, for comparison, is not COVID. Monoclonal antibody treatment, even if it's neutralizing antibody, will not be the best approach for a sure fatal disease. Once the virus gets to the nerve ending and into the nerves, it's impossible for antibodies to reach the virus, since it travels along the nerves to the brain, not blood. Rabies disease symptom is not just hydrophobia, fear for water. Victims also exhibit photophobia. Phonophobia in general, hyperexcitability. If exposure is suspected, administering antiserum may be indicated. Vaccination has to be started without delay. Now we realize why monoclonal, even if it's neutralizing against a single epitope, is not an option. Another peculiar situation is you will never know if the individual was infected or not. If they started showing symptoms, there is no turning back. If you have observed actual patient, which I have, and I don't wish to even my worst enemies, it's so sad they literally run to hide in the dark. Although there is no evidence of human-to-human transmission, their own kith and kin fear going close to them. A confirmed case of rabies recovery was reported by a procedure called Milwaukee Protocol. But the same procedure had mixed results in other situations. There are numerous factors associated with the infection. Some of them include, did the bite carry the virus? How close the bite is to the head, clothing, immune status of the individual, etc. Delayed onset even years later had been reported. It's important to discuss monoclonal therapy. COVID, it's an emergency use authorization for immunocompromised old and sick patients and certain other individuals who will not be able to immunologically respond. It's against a single epitope. Any adverse reaction, if it may happen, can be clinically managed. It's humanized, but not human monoclonal. It's still a protein and is not self. It will make anti-antibodies or anti-idiotypes. Anytime a non-self protein enters the body, it gets rejected. Fundamental basis of immune system. However, the therapy has some unintended benefits. It neutralizes the pathogen. It may even induce anaphylactic reaction. Considering the risk of COVID, it's clinically manageable. That was the basis of 1984 Nobel Prize to Niels K. Jern, who proposed idiotypic idiotypic network theory, meaning we make antibodies to pathogens, but can we make pathogen if we give antibody? It's like the dynamo and motor. Yes, we can, but not biologically active virus, but immunological parts of the pathogen. It introduced the concept of vaccine without virus, or anti-idiotypic vaccine, and has been demonstrated for rabies virus. In the paper titled Anti-Idiotypic Antibodies Induce Neutralizing Antibodies to Rabies Virus Glycoprotein. Meanwhile, Dr. Sekar Chakrabarthi from NIH will be joining me shortly. Recombinant vaccine for rabies and vaccinia as vector may actually happen. This presentation was possible with the help of Dr. Mohan's expertise in editing and computer. I sincerely thank Dr. Mohan for helping me with this computer knowledge, and the technical expertise on the subject helped this presentation.